Because a cell cannot survive long term without ATP, the amount of available ATP is closely monitored. Having too much ATP expends excessive energy in the creation of ATP, while having too little ATP puts the cell's longevity at risk. When ATP is low, it cues metabolic pathways to increase ATP production, opposed to when ATP is high, it cues metabolic pathways to downregulate ATP production. ATP production is decreased during resting conditions because ATP must be expended in order to create ATP. Within the cells, we have 4 sources of ATP. All cells have a pre-made, readily available amount of ATP. This is a small supply and only contains enough ATP to last a couple seconds. After that, we have the phosphocreatine system, as well as glycolysis, which occur within the cytoplasm. Lastly, we have oxidative phosphorylation. This occurs within the mitochondria. Try not to think about these systems working in isolation, where one begins and the others are inactive or not making any ATP. All of these systems are constantly producing ATP that can be used by the cells. What changes is the percent of ATP that they are able to contribute to total energy expenditure. One of the most efficient ways that ATP production is regulated is by the ratio of ATP to ADP present in the cell. The ratio determines the production of ATP from the metabolic pathways. In cell A, we find a high ratio of ATP to ADP. You can see the ratio of ATP is 5:2. So there is almost 3 times as much ATP compared to ADP. This is a sign that the cell is not metabolically active or has low ATP usage at this time. Therefore, it does not need to produce more ATP because it is already producing more ATP than it is using. In cell B, we see a low ATP to ADP ratio. This cell has a high metabolic activity rate right now because there is less ATP available than the cell is using. We can see that the ratio of ATP to ADP is 2:7. So the cell is almost using ATP at 3 times the rate it is producing it. It has a high metabolic activity. If cell B were to continue producing ATP at the rate it is, it would quickly use up all ATP stores and be at risk for being unable to maintain homeostasis. If the ratio of ATP to ADP drops, this means that there is an increased concentration of ADP, indicating that the cell is using up ATP because its metabolic activity has increased. An increase in ADP stimulates all 3 pathways to increase its ATP production. However, all metabolic pathways are not created equally. The speed at which ATP can be produced is directly related to the simplicity of the pathway. Some of our metabolic pathways are single-step processes, while others require more than 15 steps to create ATP. The capacity of the system is determined by the amount of enzymes we have present, as well as any training that the individual has gone through to either enhance and develop this system or limit this system. The bang for your food, this is indicated by the amount of ATP that can be created for each molecule of glucose or fat. For example, going through fast glycolysis with a glucose molecule results in only a 2 ATP production for every molecule of glucose. However, if that same molecule of glucose was able to be completely oxidized through oxidative phosphorylation, that same molecule would produce anywhere from 33 to 34 molecules of ATP. The final variable to be considered is the presence of fuel that each system can use. Glycolysis completely relies on glucose, while oxidative phosphorylations can use glucose, proteins, and fat to create ATP. As I previously stated, as the ratio of ATP to ADP decreases, it indicates that the cell is becoming more metabolically active. The increase in ADP around the cell stimulates all three systems. However, all three systems take a different amount of time to create ATP. The fastest responding metabolic pathway to the increase in ADP is the phosphocreatine system. This is an anaerobic system, meaning it does not require oxygen to complete its ATP production. It occurs in the cytosol and has a 1-to-1 ratio of ATP production for every creatine phosphate molecule. The downside to the creatine phosphate system is that there is a limited amount of creatine phosphate within the cytoplasm, as well as after creatine phosphate is used and donates its phosphate to ADP to generate ATP, the creatine molecule has to go to the mitochondria, where it utilizes an ATP molecule to recreate creatine phosphate. Therefore, if our mitochondria isn't producing enough ATP at a fast enough rate, this creatine molecule cannot be regenerated back into creatine phosphate. If we only have creatine and no creatine phosphate, this molecule no longer has a phosphate molecule to donate to ADP, which means ATP production cannot occur. The only enzyme in this reaction is creatine kinase. Creatine kinase causes creatine phosphate to donate its phosphate group to ADP to create ATP plus creatine. Since this is the only enzyme, it means that ATP can be rapidly produced, and creatine phosphate is the rate-limiting enzyme. The rate-limiting enzyme determines how fast and for how long ATP production can occur. If we are able to stimulate the rate-limiting enzyme, we are able to produce more ATP because the enzyme is more active and therefore more able to interact with more molecules of creatine phosphate and ADP. It shouldn't come as a surprise then that an increase in ADP in the cytosol stimulates creatine kinase to create ATP. Creatine kinase is inhibited by an increase in ATP in the cytosol. The creatine phosphate system can quickly reach its max output of ATP and requires several minutes to regenerate creatine phosphate. This system is best for transitional activities such as sitting to standing, jogging to running, running to sprinting, or for short 1 to 3 second bursts of activity. As I previously stated, as the ratio of ATP to ADP decreases, it indicates that the cell is becoming more metabolically active. The increase in ADP around the cell stimulates all three systems. However, all three systems take a different amount of time to create ATP. The fastest responding metabolic pathway to the increase in ADP is the phosphocreatine system. This is an anaerobic system, meaning it does not require oxygen to complete its ATP production. It occurs in the cytosol and has a 1-to-1 ratio of ATP production for every creatine phosphate molecule. The downside to the creatine phosphate system is that there is a limited amount of creatine phosphate within the cytoplasm, as well as after creatine phosphate is used and donates its phosphate to ADP to generate ATP, the creatine molecule has to go to the mitochondria where it utilizes an ATP molecule to recreate creatine phosphate. Therefore, if our mitochondria isn't producing enough ATP at a fast enough rate, this creatine molecule cannot be regenerated back into creatine phosphate. If we only have creatine and no creatine phosphate, this molecule no longer has a phosphate molecule to donate to ADP, which means ATP production cannot occur. The only enzyme in this reaction is creatine kinase. Creatine kinase causes creatine phosphate to donate its phosphate group to ADP to create ATP plus creatine. Since this is the only enzyme, it means that ATP can be rapidly produced, and creatine phosphate is the rate-limiting enzyme. The rate-limiting enzyme determines how fast and for how long ATP production can occur. If we are able to stimulate the rate-limiting enzyme, we are able to produce more ATP because the enzyme is more active and therefore more able to interact with more molecules of creatine phosphate and ADP. It shouldn't come as a surprise then that an increase in ADP in the cytosol stimulates creatine kinase to create ATP. Creatine kinase is inhibited by an increase in ATP in the cytosol. The creatine phosphate system can quickly reach its max output of ATP and requires several minutes to regenerate creatine phosphate. This system is best for transitional activities such as sitting to standing, jogging to running, running to sprinting, or for short 1 to 3 second bursts of activity. In the depiction of the cell, you can see that as ATP is used up in the resting concentrations and available ATP within the cell, more ADP becomes available for the reaction. This increases the likelihood that creatine kinase will come into contact with an ADP molecule and a creatine phosphate molecule, which increases the likelihood of ATP production. Therefore, ADP stimulates creatine kinase, increases the reactants available for the reaction, and causes an increased ATP production by the phosphocreatine system. Glycolysis is our second metabolic pathway. It can only use glucose in order to produce ATP. It is unable to utilize fats or proteins to create ATP. The skeletal muscle glucose supply can come from either the bloodstream or from skeletal muscle glycogen. Glycogen is thousands of glucose molecules bound together within the skeletal muscle. The storage of glucose as glycogen serves several purposes within the cell. First, it allows for a large amount of glucose to be located close to where glycolytic pathways are most prevalent in the skeletal muscle. In addition, storing glucose as glycogen allows the maintenance of the glucose concentration gradients. If the skeletal muscle stored glucose, it may result in skeletal muscle having more glucose inside the skeletal muscle than in the bloodstream. This would result in passive transport of glucose out of the skeletal muscle into the bloodstream, or require active transport to get glucose into the skeletal muscle. Glycogen also prevents osmosis and the movement of plasma from the bloodstream into the skeletal muscle. Water moves towards areas of high concentration of molecules compared to low concentration of molecules in order to maintain equal dilution. If glycogen was not formed, thousands of molecules of glucose would be inside the cell, creating a high concentration of molecules and therefore a high pull of water out of the bloodstream and into the skeletal muscle. This could result in dehydration or even the plasma membranes bursting from too much water being inside. However, when two glucose molecules are bound together, it results in one molecule. This decreases the concentration of molecules and prevents water from entering into the cell. As the cytosol ADP concentration increase, it also starts to stimulate the process of glycoglenolysis. This is simply saying that we're breaking down glycogen into its subunits of glucose. As glucose is broken down, it can then be used for glycolysis. Glycolysis is a prime example of why we don't want to be creating ATP unless we're also using it, because in order to prime glucose, in order to put it through glycolysis, we have to invest ATP. The first cost of ATP comes from turning glucose into glucose 6-phosphate. Following this, glucose 6-phosphate is turned into fructose 6-phosphate, and the enzyme PFK turns fructose 6-phosphate into fructose 1,6-phosphate. The important information for you to remember here is that it costs ATP to go from glucose to glucose 6-phosphate, and it also costs 1 ATP to go to fructose 6-phosphate to fructose 1,6-phosphate. The last point of emphasis here is to remember that the enzyme PFK is in charge of the reaction between fructose 6-phosphate and fructose 1,6-phosphate. This will come back later when we talk about rate-limiting enzymes for glycolysis. At the end of the priming stage, we are left with one molecule of fructose 1,6-biphosphate. As we begin the harvesting phase and collecting the gains of glycolysis, we start by splitting fructose 1,6-phosphate into two 3-carbon molecules. G3P and DHAP are the resulting 3-carbon molecules, and as we convert these into 1,3-biphosphoglycerate, or BPG, we are also going to net 2 NADHs and a resulting 2 hydrogen ions. As we convert 1,3-biphosphoglycerate into a 3-phosphoglycerate, we also convert 2 ADP into 2 ATP. In the final step, converting phosphoethanol pyruvate or PEP into 2 pyruvate molecules, we also net the last ATP. The important components to remember in this harvesting and gain site is that the 1 6-carbon molecule of glucose is eventually converted down into 2 3-carbon molecules. And here on out, we're talking about 2 molecules instead of 1. It is also important to remember that as we convert from those 3 carbon molecules into 1,3-biphosphoglycerate, we're netting 2 NADHs and 2 hydrogen ions. It's also important to know that as we convert from 1,3-biphosphoglycerate to 3-phosphoglycerate, we are adding 2 ATP to our ATP production. And also as we are converting between steps 9 and 10, or between PEP and pyruvate, we add the last 2 ATP molecules. In total, when glucose goes through glycolysis, we have 2 different options for net gains depending on where the source of the glucose was from. If the glucose came from the blood, we had to prime glucose into glucose 6-phosphate. However, if the glucose came from muscle glycogen, we did not have to prime glucose into glucose 6-phosphate. Therefore, muscle glycogen going through glycolysis gives us 1 additional ATP per molecule of glucose compared to glucose brought in from the blood. In terms of gains, both glucose molecules have the same gain. We gain 4 ATP, 2 NADH, and 2 hydrogen molecules, along with 2 pyruvate molecules. In sum, our net gain, if blood glucose is utilized through glycolysis, is 2 ATP, 2 NADH plus the 2 hydrogen ions, and 2 pyruvate molecules. If the glucose came from muscle glycogen, we net 3 ATP molecules, 2 NADH molecules plus 2 hydrogen ions, and pyruvate. In option 2, or fast glycolysis, the pyruvate is either unable to enter into the mitochondria for some reason, or the NADH is unable to shuttle its hydrogen ions into the electron transport chain. However, we need NADH in order to continue glycolysis and ATP production. Therefore, pyruvate interacts with lactate dehydrogenase. The NADH and hydrogen ions are converted into NAD and lactate. Lactate helps absorb those additional hydrogen ions, and this process helps recycle NADH back to NAD, continue the glycolytic pathway. Understanding how we control the rate of glycolysis is an important topic for exercise physiologists. The rate-limiting enzyme is phosphofructose kinase, which is found during the investment stage of glycolysis. Often, the rate-limiting enzyme is found early in the metabolic pathway because it prevents the accumulation of products later in the cycle. Phosphofructose kinase, or PFK, is stimulated by an increased concentration of ADP in the cytosol. Again, an increased concentration of ADP indicates that there is inadequate ATP production for the metabolic demands of the cell. By having PFK be stimulated by ADP concentrations, it activates PFK to increase its metabolic rate and lead to the greater production of ATP. Conversely, PFK is inhibited by an increased concentration of ATP, which would indicate a lower metabolic demand of the cell, an increase in hydrogen ions, which can decrease the pH of the cell and limit enzyme activity, and an increase in citrate. On the previous slide, we talked about how pyruvate can enter into the mitochondria during slow glycolysis and is converted into acetyl-CoA to enter into the Krebs cycle. Acetyl-CoA is then converted into citrate. If there's an increase in citrate, that means there's a backup of byproduct in the mitochondria, and if PFK continues to create pyruvate, it will be immediately turned into lactate. Another control of glycolysis is phosphorylase. Phosphorylase is the enzyme that breaks glycogen into glucose. Phosphorylase is stimulated by an increased concentration of cytosol calcium and an increase in epinephrine. We see an increase in calcium through the increase of muscle contraction of a muscle fiber, and we see an increase in epinephrine with sympathetic stimulation, often found in exercise. By having phosphorylase be stimulated, it will break down more glycogen into glucose, which allows more glucose to enter into glycolysis. To summarize, fast glycolysis is only able to work if it has available glucose from either the bloodstream or skeletal muscle glycogen. If the glucose is extracted from glycogen, 3 ATPs per molecule of glucose is formed. If the glucose comes from blood glucose stores, 2 molecules of ATP are formed for every molecule of glucose. Fast glycolysis has a large capacity and is able to increase its rate of ATP production at least 1,000 times. It's ideal for slightly longer durations at high intensity compared to the creatine phosphate system. All of the reactions occur within the cytosol, and it is an anaerobic metabolic system, meaning it doesn't need oxygen to function. The end product is lactate and a regeneration of NAD, which allows glycolysis to continue. Its rate-limiting enzyme is going to be phosphofructose kinase, or PFK, and in slow glycolysis, the pyruvate is shuttled into the mitochondria and is converted to acetyl-CoA. The production of acetyl-CoA releases carbon dioxide and electrons. Each pyruvate results in 1 acetyl-CoA, and each acetyl-CoA turns the citric acid cycle 1 time. Therefore, at the end of the citric acid cycle, we need to multiply everything by 2 in order to determine how much energy 1 glucose molecule can make, because 1 glucose molecule created 2 pyruvate molecules. Acetyl-CoA combines with acetylacetate to form citrate. What results next is a series of oxidation and reduction reactions. Very little ATP is formed during the citric acid cycle. In fact, only 1 ATP molecule is formed for every turn of the citric acid cycle. And this occurs between succinyl-CoA and succinate. In this step, we produce a GTP. That GTP is a high energy molecule, which then donates its third phosphate to ADP to create ATP and regenerates itself as ADP to continue on the citric acid cycle. The other key steps of the citric acid cycle include in between isolocitrate and ketoglutarate formation. During this step, we form another NADH. In between ketoglutarate and succinyl-CoA, we form another NADH and carbon dioxide. And finally, in the conversion of maltate to oxaloacetate, we form our last NADH molecule. Each citric acid cycle creates 1 FADH in the conversion between succinate and fumarate. Each NADH and FADH has 2 electrons attached to it. As you can see, the citric acid cycle does not result in much ATP formation, but results in significant NADH and FADH. These molecules are then able to shuttle the hydrogen ions to the electron transport chain, where they can then be used to make larger quantities of ATP. The rate-limiting enzyme to the citric acid cycle is isocitrate dehydrogenase. Again, as probably is becoming pretty redundant, an increase in ADP results in the stimulation of this enzyme. Again, the increase of ADP indicates that the cell is metabolically active and expending more ATP than it is producing, and therefore requires an increase in ATP production. Also, an increase in cytosol calcium results in an increased activation of isocitrate dehydrogenase. Again, calcium concentration increases as muscle contractions occur. If we're going to talk about something that inhibits isocitrate dehydrogenase, we would look at the opposite of a metabolically active tissue and realize that if ATP rates were high, the tissue is probably inactive and does not need more ATP production. Therefore, ATP being increased results in the inhibition of isocitrate dehydrogenase. Depending on the intensity of the exercise and the presence of oxygen, the destiny of glucose can follow one of 2 paths. The first would be fast glycolysis, where we end up with 2 pyruvates and 2 NADH plus hydrogens. This is converted by lactate dehydrogenase into 2 lactate molecules and NAD positive. There is a net gain of 2 or 3 ATP depending on where the glucose came from, either the blood or glycogen. The other option for glucose is to go through slow glycolysis, where more of the glucose molecule is oxidized and reduced. The 2 pyruvate molecules at the end of fast glycolysis, instead of getting converted into lactate, are shuttled into the mitochondria. In the conversion of pyruvate to acetyl-CoA, 2 NADHs are formed. Each of the acetyl-CoA molecules that are formed will turn the citric acid cycle once. So 1 molecule of glucose results in 2 turns of the citric acid cycle. The net of these turns are 6 NADHs, 2 ATP, and 2 FADH. If we add together the products from fast glycolysis, we would end up with 10 NADHs, 2 or 3 ATP, depending on where the glucose came from, and then the additional 2 ATP and 2 FADH. The electron transport chain utilizes the hydrogen ions from the Krebs cycle to create a concentration gradient between the mitochondrial matrix and the inner mitochondrial membrane. It creates a high concentration of hydrogen ions in the inner mitochondrial membrane compared to the mitochondrial matrix. This gradient, when allowed to flow in its natural passive direction, can do work. Gradients want to move from high concentrations to low concentrations and do so at a faster rate the larger the difference between the sides of the gradient are. This gradient is very large, and the gradient is used to add phosphate to ADP to form ATP in water at the final pump of the electron transport chain. There are 4 pumps within the electron transport chain for hydrogen to enter and to utilize the gradient to do the final electron acceptor. The electron transport chain consists of 4 different cytochrome pumps. NADH drops its hydrogen ions at pump 1, while FADH enters its hydrogen ions at pump 2. Pump 4 is the last pump where hydrogen ions are released and combined with oxygen, ADP, and phosphate to form ATP and water. If no oxygen is present or insufficient oxygen is present, the electron transport chain cannot continue because there is no final electron acceptor, the final hydrogen ions that are being released from the inner mitochondrial membrane into the mitochondrial matrix. If sufficient oxygen is present, the electron transport chain can continue to use its concentration gradient to add hydrogen onto oxygen to form water and ATP. Since NADH drops its electrons off earlier at pump 1, it is able to produce 2.5 ATP molecules per molecule of NADH. Since FADH drops its hydrogen ions off at pump 2, it is only able to create 1.5 ATP molecules per FADH molecule created. If we compare the outcome of one glucose molecule through fast glycolysis or slow glycolysis, we can see there's a definite increase in the amount of ATP generation if we are able to fully oxidize and reduce glucose. Through fast glycolysis, 2 or 3 ATP are generated along with 2 lactate molecules and the regeneration of 2 NAD positive molecules. If we are able to shuttle that pyruvate into the mitochondria and put it through the citric acid cycle, not only do we end up with the 2 or 3 ATP, 2 NADH that are generated during fast glycolysis, we end up with an additional 8 NADHs, FADH, and 2 ATP. Since we know that NADH is able to enter the electron transport chain earlier than FADH, which results in a greater amount of ATP production per molecule, we can see that the end result of 10 NADHs will give us 25 molecules of ATP, 2 FADHs will give us 3 molecules of ATP, and then 4 or 5 additional ATP molecules depending on the origin of the glucose. If we are able to fully oxidize a glucose molecule through slow glycolysis, we are able to net 33 or 32 ATP molecules compared to the 2 ATP molecules from a glucose if we go through fast glycolysis.